Good morning, students. This is the last lecture in our series of Bio 102, and we are going to end the entire adventure with plants. And it's just going to be a very brief introduction on plants. We'll just scratch the surface of this topic because it's a really huge topic, but we just want to know just a little bit about plants. And so we'll start out by saying that plants, of course, are multicellular eukaryotes. And they form a clade that includes angiosperms, which are flowering plants, gymnosperms, which are plants that don't flower. Gymno means hidden, so hidden flowers, ferns, mosses, and green algae. So these are all bunched together in a clade. Red and brown algae are actually excluded from green plants, even though they're algae, as of course are all animals and fungus. So that is what a plant would be when we talk about plants in the next few minutes. Remembering again from the tree of life, where are exactly plants? So we start out at the base of the tree of life, and that would be the last universal common ancestor, or Luca, who we don't know who that was or exactly what time that was, but it was very early on, about 4 billion years ago, approximately, roundabout. And that simple organism then evolved into these three big domains of all life. The domain in blue, this one, became bacteria. And that domain was very different from this other domain, which was the original Luca. And that domain then broke up into two other domains, which are archaea, which are full of the extremophiles, and then eukarya, which is everything else, which are animals, plants, fungus, and everything, the little single-celled organisms that are eukaryotes. So that's just a review of where plants stand and how we're connected to them. All green plants have many features in common, including cell walls containing it should just be containing cellulose, as they all synthesize their own food by using chloroplasts and sunlight, and they store food in the form of starch. So this is common throughout this plant kingdom. Plants are also characterized by indeterminate growth. Now, what exactly does that mean? That means that they can grow for an indeterminate time. That means there is no real fixed life or death span for them. Some plants can live for a very, very, very long time, depending on good conditions, and some plants could live just a few months. And plants also have asexual reproduction as well as sexual reproduction, and they have an alternation of generations. So this is kind of cool because these are their features. The earliest plants were stromatolites. So the undisputed evidence that we have of these plants is 3.5 billion years ago. We believe that they were there before that too, but stromatolites were the earliest algae, and they gave rise to all the plants that we see today. So the earliest algal mats that arose from the stromatolites that gave rise to this plethora of plants that we see today, that actually started about 450 million years ago. So all the plants that we see on land are not that old compared to the age of the Earth, because the age of the Earth is 4.6 billion years. And all these plants that we see are not even 500 million years old. So and the early plants, the stromatolites, they have long since become extinct, but from them arose other simpler plants. Photosynthesis originated about 1,000 million years ago. And just so you remember that one million is a billion. So photosynthesis was a new invention. And embryophytes or land plants include the vascular plants. So land plants are different from the plants that are in the water. Land plants include ferns and conifers. Those would be the gymnosperms. And then flowering plants, which are called angiosperms, and also include these very primitive non-vascular bryophytes, commonly known as mosses and liverworts. So these guys are very, very primitive and considered very old. So if you were to make a diagram of how they evolved, you would start out, of course, with the embryophytes, which are the land plants, which divide into two branches, vascular and then non-vascular. In the non-vascular, those are the very ancient ones called bryophytes, and they evolved into things called mosses and liverworts and hornworts and very ancient plants. However, the vascular plants then branched off into two. They became ferns, which was another invention, and then others decided they would have seeds, and they became seeded plants. Out of the seeded plants, there were two new inventions that came out. One was no flowers or gymnosperms, and one was flowering plants. So that is how we can trace all our plants today to backwards. If we wanted to go backwards to their ancestors, that's how we would do that. Here you see the same idea, and what we are looking at over here is if you look on the right-hand side, you'll see vascular plants, and that would be this bar, okay? And it starts here and then it goes up to here. But land plants or embryophyta, and they go up to here. So this would be up till here. Before land plants, we have streptophyta, and they go up to way back. And then before that, we have chlorophyta, which actually had the chlorophyll. So this is how we would divide it all up. And you see examples of flowering plants. So these are at the apex. And then the gymnosperms here, the ferns here, the mosses and so on are lower down. So these are older plants, older inventions. And then, of course, the algae and so on are even way down back here. Here is another way of looking at it. We like to look at things bottom up. And so when plants started to come to the land, this is what happened. They developed into branches. So we have a branch called liverworts. They also developed into hornworts. Sometime later, they developed into mosses. Mosses we still have today. And mosses branched out. At this fork in the road, there was a choice, either become a moss or a vascular plant. And the vascular plants have, again, two branches. One is club mosses, but the other branch, which is more significant, are the ferns and the seeded plants. And from the seeded plants, you have the flowering plants and so on. So it's the same idea, but just giving you a different perspective of this. So we should start out with plants that are very, very ancient and primitive. So those would be bryophytes. These are non-vascular plants, and they have an alternation of generation. So that means the haploid stage, where the plant actually just has half the DNA component, or is just a gametophyte, looks like a fully fledged plant, and it alternates with a diploid stage called the sporophyte. So what you see here are the plants in a haploid form, but they look like real plants. So this is a very common thing in the plant kingdom. What you see in pink is how long the plant spends as a diploid organism. And the teal colored branches that are here and here, these show you the plant living in an haploid form. So we can, since it's a circle, we can start anywhere. We can start here where the organism starts to become diploid. There's fertilization. There's a zygote. The zygote becomes an embryo. And the embryo now is diploid. And it becomes actually a plant. The plant gives rise to female gametophytes, and they become mature spores, which are dispersed. So they have two kinds of spores, male and female, and what you have is a whole life cycle living as a female haploid, or you could have a whole life cycle living as a male haploid plant. And the only way you could tell they're different is when you actually see what do they produce. The haploid female plant is going to produce eggs, whereas the haploid male plant will actually have anthers, which will produce spores, which will be carried by rain to the female plant, which will fertilize the egg, and then the cycle begins again. So that is how bryophytes would work. Here you see plant groups. So let's look at this a little bit carefully. So we just looked at this example a minute ago, and that's fine. So let's check and see what are the dominant parts of the life cycle in plants. Is it haploid or diploid? In mosses and other non-vascular plants, of course, we saw that the dominant part of their lifestyle is to be a gamete, which is haploid. And so the sporophyte, which is actually going to become a real plant because it has the full component of the DNA, is a very short amount of time. And as you can see, the sporophyte is diploid, but it forms haploid male and female gametes. And it lives for a short amount of time. Moving up the scale, if you see ferns and other seedless vascular plants, the gametophyte is not the biggest part of its life cycle. It is independent, it is photosynthetic, it is free living. But the sporophyte, or the two-end part, is what we generally see in nature. The very pretty ferns, they are actually diploid. The haploid part is the gametophyte, and we don't see that much. What we remember in ferns is its pretty leaves. Moving up the complexities style, we could go up to seed plants, which are gymnosperms and angiosperms. So gymnosperms do not make flowers, and angiosperms do make flowers. Here, the gametophyte or haploid stage is usually pretty small. It's just microscopic. And the two-end stage or diploid stage is the stage that we actually see. And what you see over here, so this is now branched into gymnosperms and angiosperms. So gymnosperms, of course, do not make flowers. And what you see is in conifers, the cones that you see yourself under the branches, they have the haploid eggs. And then you will see these fuzzy-looking things too on the same plant, which will have the male gametes, but the entire tree is diploid, just like us. In angiosperms, what you would see is the plant, and then the plant will have two parts where it is making just haploid gametes in the flower. So the flower will have a part where it just makes eggs, and then a part where it just makes sperm, but the entire plant is diploid. So this is another way of representing the alternation of generations in vascular and non-vascular plants, and this actually shows you the increasing complexity. So when we start out with green algae right here, they spend most of their time haploid. So their entire life cycle is spent haploid, and that's depicted by how big that is shown. The diploid part is very small, and that's just the zygote. When you go on to bryophytes, they do spend a considerable amount of their time as haploid, but as you can see now, the distance here is getting bigger, so they do spend some time as a diploid organism. When you go on to ferns, which are more complex than bryophytes, they spend most of their time as a diploid organism. We do see that, and the haploid part we don't see that much, but that is getting now smaller. And then when we go to gymnosperms, you can see that now if you drew this all the way to the y-axis, well, and draw it perpendicular to the x-axis, the plant lives most of his life diploid, and the haploid part is just reduced to the sperm and the eggs. And then angiosperms, which are the most advanced plants that we have, they spend all their lives diploid, and very little part is spent haploid, and that is only the gametes. Vascular plants developed a number of adaptations like vascular tissues to transport water and food, and root systems capable of obtaining soil water and nutrients. And this was a necessity because plants had originally started out in the oceans. When they came to land, they needed to adapt to the land where water is no longer plentiful. So they needed to transport that water and to obtain it. In vascular plants, the sporophyte, which is the 2N part, so 2N, diploid part of their lives, is nutritionally independent, and it's long-lived, and the gametophyte phase has evolved to become really, really reduced. Vascular plants, again, include ferns, gymnosperms, and angiosperms. So when we look at the seedless vascular plants, like for instance ferns, this is their life cycle. So the sporophyte, or the 2N part, is large and long-lived, but when young, it actually depends on the gametophyte for nutrition. The spores are produced. If you ever turn this the fern leaf over, you'll see these little brown dots at the base of their at the bottom of their leaves. And in those dots are actually spores, which are dispersed by the winds, and they develop into gametophytes, which are develop into little plants, which we don't really see that much because these are showy, and so we see the big showy ferns instead. These are very small, and they kind of stick around to the ground. These gametophytes will develop into eggs or sperm. The sperm are going to swim to the egg and fertilize it, and then you get a gametophyte developing right here that is now a zygote. And that zygote will then develop into a full-fledged plant, but notice there's still food sitting around for the baby diploid organism to get started out. There are 3 kinds of cells in plants, and we're going to look at all of them to see what makes the diversity of all the plants that we look at. It actually comes down to 3 types of cells. Just like we do, we just have 3 types of cells too. Not that complicated, but we can then smush them or push them around and make them into new shapes and come up with the variety that we do see. The 3 types of cells that plants carry are called parenchyma, collenchyma, and sclerenchyma. Parenchyma cells form the bulk of non-woody plants. They're loosely packed, they're cube-shaped or elongate cells with a large central vacuole and thin cell walls. They are the most abundant and the least specialized cells, but they can specialize for various functions. So parenchyma is like the catch-all cell. It can become something else. It's loosely packed, it's cube-shaped, it's got a big old central vacuole in it to control the water intake, and they're the most abundant cells that are in a plant. The collenchyma cells are irregular in shape. They have thicker cell walls. They're flexible enough to support the growing parts of plants. But the sclerenchyma cells are the really thick, rigid ones. They support the non-growing parts of the cells of the plants because they have thick, rigid, and non-stretchable cell walls. And so those replace the collenchyma as the plant grows. There are two types of sclerenchyma cells called fibers or scleroids. Fiber cells are about 50 centimeters long. They usually occur in strands, but scleroids have thicker cell walls. And we're going to take a look at them. So this is what a parenchyma looks like. So as you can see, they're all kind of, irregular shaped, loosely packed, not squashed together. Collenchyma, you can see, are bigger, so they give structural support as long as the plant is green. And then the sclerenchyma cells, they have these fibers in, and they will they become these rigid bodies, so they can support the plant to stand up. Plant tissues make up the main organs of a plant. So the main organs of a plant would be root, stem, leaf, and flower. And there are three types of tissues we just thought about. The three types of cells become three types of tissues, and they're continuous in the entire plant. So we have ground tissue that makes up most of the plant's body. Then we have dermal tissue, which is going to cover the outside of the plant. And then there's vascular tissue, which conducts water and nutrients. And this makes a lot of sense, because the plants are not very complicated, and all they really have are three types of tissues. The dermal tissue, which is the easiest one to describe, is just the outside of the plant, and that would be the epidermis. Usually there's a cuticle attached to it, so it's kind of a little bit to reduce water loss. So there's some mechanism for that. And the vascular tissue, which is for transport, has two parts to it, xylem and phloem. Xylem is for transport of water, and phloem is for transport of nutrients. And then there's the ground tissue, and that's going to be something that's going to fill in between these two. So between the epidermis and the vascular, there's got to be some filler, and that is called the ground tissue. That is the bulk of the plant. If it's in the center of the plant, it's called pith. If it's external to the vascular tissues, it's called cortex, and it's used for photosynthesis, for storage, and for support. The dermal tissue covers the plant body and consists of the epidermis in young plants, and it's later replaced by periderm in woody plants. So periderm is actually the wood, whereas epidermis would be nice and green. Epidermis is made of parenchyma cells, but they're in a single layer. Epidermis on stem and leaves prevents water loss by transpiration and produces a waxy material called cuticle. Epidermal cells on roots form extensions called root hairs to absorb water and nutrients as they go seek it. Openings in the epidermis on the underside of a leaf are where gases are exchanged, and those are called stomata. Stomata are these little holes, and they have two cells around them that are sausage-shaped, and those are called guard cells. So these are the sausage-shaped guard cells that I'm drawing kind of not so well. And that would be where the gases are exchanged because that is the hole. And the stomata actually control how big is the hole going to be during the day and night, and that will control how much water is going to be lost or conserved. And this is an actual picture of a stomata and two guard cells. So here's one guard cell, And here is the other guard cell. And you can see that this is actually the opening where the plant connects with the environment. Ground tissue constitutes the majority of the plant body and contains parenchyma, collenchyma, and sclerenchyma cells. Ground tissue of the leaf is called mesophyll, and we did look at mesophyll because we said that's where the chloroplasts are. The mesophyll uses the energy in the sunlight to synthesize sugars, or does photosynthesis. Ground tissue of the stem is called pith and cortex, and that develops support cells to hold the young plant upright. Ground tissue of the root is also called the cortex. It often stores energy-rich carbohydrates. The vascular tissue, which is very interesting. The vascular tissue has two parts. There's xylem and phloem, and you need to remember that the xylem transports water and minerals from the roots to the leaves and the shoot. So in xylem, food, water is going up along with minerals, and they're going up from the roots all the way to the top of the tallest leaf. Phloem, however, it goes in the opposite direction. So stuff in it is actually food. So the leaves synthesize food, and so phloem is the channel that's going to transport that from the leaves to the rest of the plant and to storage facilities wherever the plant stores its extra food. Xylem has two kinds of conducting cells. They're called tracheids and vessel elements. Tracheids are long, narrow sclerenchyma cells with walls and pits for water to move between them. And I'm going to show you a picture of them. And vessel elements are short, wide sclerenchyma cells without end walls stacked on top of each other. Angiosperms have tracheids and vessel elements, but gymnosperms only have tracheids. So I want you to kind of look at this. Here we have a picture of xylem cells. All right, so let's take a look at it. This dark brown thing is called a tracheid, all right, and you can see little pits in it. And so the water is going to move from the roots all the way up to the shoot, and it's going to move between tracheids only through the pits, these little holes. Right, so what you see is besides tracheids, what you're also going to see is vessel elements, and from vessel elements, in vessel elements you actually see perforations. So water moves from one vessel element to the next vessel element through perforations, which are these pits, pits and perforations. So the perforations are bigger holes. So water is moving through these two types of holes in the xylem to get to the leaves. Phloem is made out of cells called sieve tube members and companion cells. Sieve tube members are stacked to form tubes called sieve tubes with porous sieve plates between the cells for the movement of sugars. Companion cells are along each sieve tube member and help in loading sugar into the sieve tube. As their name would say, they are companions, so they help along. And this is what a phloem cell looks like. So let's look at it. This is different from a xylem, and what you see is a nucleus. So here's a cell, and that's the nucleus. Here's a companion cell, but it has a sieve plate. A sieve plate would have holes in it. It also has pores. And the cytoplasm of adjacent phloem cells are connected through tiny pores in their cell walls. So food can move this way. It can also move this way. So that's how phloem will move the stuff around. So let's see how water moves in xylem and phloem. On the left-hand side is xylem. On the right-hand side is phloem. And they're both tubes, but they both do different functions and they both act differently. So if you look at sugars in the phloem, like here, sugars are actively transported from source cells into sieve tube elements. So they are going to be moved down. So here are the sugars. So they're moved from the source cells into the actual tube itself. Because of the high sugar concentration in the phloem, water is going to have to move in too, yes? It's going to diffuse into the sieve two elements, and once water moves in, then the water pressure rises. And because the pressure has now risen, this causes the sap sugar water to flow through the phloem, and sugars are transported out of the phloem into sink cells. So this would be a sink cell, like where they want to store this sugar. The water will then diffuse into the xylem, and it will reduce the water pressure into the phloem. So let's say that this is a source cell in a leaf. The leaf makes the sugar goes down, 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 down into the root, where it is then stored in some sort of sink cell. And when that happens, the sugar is moved out, while the water is just going to go back up into the xylem, and it's going to be reused to make food again. So we don't have to keep on slurping up water if we're a plant, because we're just going to simply reuse it again and again and again. There is a cohesion theory of water movement. Transport of water and mineral nutrients occurs in the xylem, and we must remember this. Water movement in plants is driven by transpiration. Transpiration is evaporation of water from the leaves and stems. As the water evaporates, more water is pulled into the roots. So that's because of the property of cohesion of water molecules. Transpiration will produce a negative pressure in the xylem, pulling the water upward. And water molecules, as we know, are cohesive, which means they're attracted to each other, and that will also pull the water upward. Water is also adhesive, which means it sticks to the walls of the xylem, so that helps to pull the water up. And so here we're going to look at the entire plant, and we're going to look at all the parts of the plant. So let's just look at one generic plant. This line demarcates the boundary between the soil and the air. Under this line, whatever is under is the root. There's usually a primary root, and there are branches coming off of it called lateral roots. When this root goes out of the soil and into the air, it becomes a stem. It's called a stem, and the stem is different completely from the root, and it's usually green, and it carries a lot of leaves. And these leaves are usually green because they carry on photosynthesis. So let's look at the functions of the shoot system, which is anything that is above ground. Its function would be, of course, to do photosynthesis, which is make food. Its function would also then be to store the food or transport the food. But its function is also reproduction, because here is where all the reproduction is going to go on. And it is also controlled by hormones. Believe it or not, plants do have hormones. And then in the roots, what are the functions of the roots? Well, first of all, it's got to anchor the plant. That's really important. The next really important thing is it's got to absorb some water, because that's very important. Got to bring the water in and nutrients in, and then it also will store extra food. It will also have to transport that water and food around, and then also is controlled by hormones. Let's look at the names of the stem parts. So something between two leaves is called an internode. That means internode means there is nothing growing there. There are no leaves there. But at every node you will see something growing, and that node in this case happens to have a leaf. Tucked in between the leaf node and the stem is usually a bud. And that bud is an axillary bud because it's on the side. But if the stem ends in a bud, that's called an apical bud, because it's at the apex. Roots are non-leaf bearing, non-node bearing parts of the plant, and they're usually below the soil surface. And they can be aerial. So there are some plants that are aerial, and they perch on other plant branches, and they just suck up any moisture from the air, but for the most part, roots are usually below the soil. There are structural and functional differences between roots and shoots, and we're going to look at that. Roots absorb water and inorganic nutrients. They anchor the plant, they support it, they store food, and they do vegetative reproduction. Some roots can actually run around and produce more plants as they run around. We'll look at that. Roots can be fibrous, as in monocotyledons, or tap, as in dicotyledons, and we're going to look at what a monocot and a dicot is. But let's look at a root structure first. So here is a generic root, and here is it's growing downwards. It has lateral branches. That's the primary root, this going down, and the little fuzzy things that you see coming out are root hairs. This is the root tip, which is capped, and this is where the root elongates or gets longer and longer. This area is called the meristematic zone, where things are constantly being built or getting bigger because it's trying to go get more food. This is the zone of maturation, where it's going to get wider this way because it's already established, and now it just needs to put down food and make itself wider. So let's look at the structure from the outside in. If we cut the root, what do we see? So on the outside, there will be root hairs. We just looked at those fuzzy things. Then there's going to be the epidermis. And then there's going to be cortex. And then there's an endodermis, a pericycle, and then the vascular tissue that's in the center. Root growth starts from the apical meristem, which divides continuously, and it grows down in response to gravity. The apical meristem produces cells that elongate, which pushes the root tip forward, and that's called primary growth. Here's a picture of what I was just talking about. So here are the root hairs. Okay, they're outgrowths of the epidermis. These are the epidermis cells. And then this filler stuff is all cortex. Cortex will just, hold starch or extra food or serve for anchorage and maybe have water in it if there's a need for extra water to be stored over there. Then what you notice is that this actually surrounds something that is circular. The circular bundle is bound by something called the endodermis, which is a layer of a single layer of cells, which binds something called the pericycle. The pericycle is very important. We're going to look at that in a minute. And what you see then are two distinct areas, these little areas that are in between some arms. That is the phloem. And the star-shaped thing that you see in the center is xylem. So the xylem is really pronounced in the root because it has to bring in food, water, and nutrients. So it's rather big. And so this is the vascular tissue, and there's cambium over here, which is tissue that's going to differentiate into xylem and phloem. Here is, so again, let's look at the meristems. Remember we talked about the apical meristem at the apex of a shoot, and that is where the elongation of the shoot occurs upwards. But there's also an apical meristem at the bottom of the root, and that's where the elongation of the root occurs. And we also noticed that there were axillary meristems, which are just going sideways. And when we actually look at what a meristem looks like, let's take a look at it. So here, if you look at this, these rows and rows of cells, that is the apical meristem. They are covered loosely by something called a root cap, which protects them. These are actively growing cells. They keep dividing. They have a cover of other cells which keep breaking off as the root pushes forward. So here is another picture of the same idea, but now it's in color. So here's the root cap. Here's the apical meristem, the growing part, and root cells are going to roots are get elongated by both cell division and elongation. So the root grows in length as well as in width. And as we, keep on going up the root, you'll see root hairs coming out on the sides, and the xylem and the phloem. So the phloem is in blue, so that is here, and then the xylem is given in red, and this tissue around here is just simply the endodermis, which is going to line the pericycle, and the pericycle binds the vascular bundle. So 3 types of meristems, we need to remember what they are. Apical meristems are those that are at the apex of a plant. So they can be either the stem or the root, and their function is growth. They increase the length at tips. The intercalary meristem, this is the meristem between the tip and the base of the stems and leaves, and they are going to increase the length between the nodes, so their function is also growth. And then the lateral meristems, they're on the sides of stems and roots, and their function is also growth, and they increase the diameter of the plant. The root cap covers the apical meristem, which is the growth tissue at the tip of the root, and produces a slimy substance so roots can more easily grow through the ground. The apical meristem replaces cells of the root cap as they are damaged, as they're being pushed into the soil. The core of the root is called the vascular cylinder, and that contains the xylem and the phloem. A band of ground tissue called cortex surrounds the vascular cylinder. A single cell layer called the endodermis separates the cortex and the vascular tissue. Endodermal cells are coated with a waxy layer called the Casparian strip. And so water is channeled into the vascular tissue. The whole function of the wax is so the water doesn't move the other way, but it moves towards the center. The pericycle is the outermost layer central vascular tissue, and it forms the lateral roots. Let's see how it works. So this is the root structure. So again, where we see the root hairs. So this comes from the epidermis. And then what we see is the cortex. So all this stuff is just cortex. But then what we are also looking at is this one layer of endodermis, and it's given in blue, which surrounds a layer in orange, which is the pericycle. The pericycle is where the root hairs actually start to go this way. And this is all coated with the wax, so the water doesn't go this way, but actually goes this way. And this would be the xylem, and of course this is the phloem. Secondary growth begins when the vascular cambium forms between xylem and phloem. And this encompasses growth in diameter. So it occurs at lateral meristems, and you can get cork cambium and vascular cambium. Vascular cambium forms secondary xylem and secondary phloem, and cork cambium produces periderm, or just cork. So plants with secondary growth have a cylinder of tissue along the stem and the root. The vascular cambium forms xylem on the inside and phloem on the outside. And vascular cambium produces secondary xylem towards the inside of the root and secondary phloem towards the outside of the root. So here's a picture showing you movement of water in a root. So here is a root hair, okay? And then what we see is water droplets, all right? And what we're seeing is water and minerals are going to enter the root hairs by osmosis. And so the water direction is this way. And here we are, we're finally at the endodermis, and we get to the waterproof seal. Once water gets here, notice this is all cortex, and then this is the endodermis, one layer, and that's the pericycle. And then that's the phloem, and that's the xylem, and so water is going to go all the way into the xylem like that. Springwood forms in spring when there's lots of water and xylem cells are wide with thin walls. Summerwood forms when there's less water and smaller xylem cells with thick walls form. In the stem cross-section, the abrupt change between small summerwood cells and the following year's large springwood cells produces an annual ring. So you'll see that when the tree is cut down, you'll see these annual rings, and they're formed only because of the size of the cells due to how much water they're absorbing. Moving on to the leaf, where all the action happens. The flat surface of the leaf helps capture sunlight for photosynthesis. It converts carbon dioxide and water into simple sugars like glucose. Some leaves store food, like the onion leaves, and they store water too. Some leaves are used for protection, as in cactus spines. Leaves are attached by a stem-like petiole to the plant. So there's a little attachment so that the leaf can furl out and do photosynthesis. Simple leaves usually have one blade. Compound leaves have several leaflets. We're going to look at that in a minute in pictures. The leaf is covered with a single layer of cells called the epidermis. There's an upper epidermis and a lower epidermis, and there's a waxy cuticle on top of this which prevents water loss. And there are openings called stomata on the underside of leaves for gas exchange, so carbon dioxide and oxygen are pulled in and out. Oxygen is sent out and carbon dioxide is sucked in. Two guard cells on either side of the stomata will open and close the openings. When guard cells lose water, the stoma close, while the stoma open when the guard cell gains water and it swells. So this is a very clever way of controlling the opening of the leaf to control water. So here is the guard cell, and if it's turgid, this will open so more water can leave. But if it doesn't have a lot of water, it's going to actually close up that, so not any water is going to be leaving the leaf. Stomata are closed during the hottest parts of the day to prevent water loss from the leaves. So these open and close cyclically during the day. Below the epidermis are two types of chlorophyll containing mesophyll cells, and they're called palisade and spongy. Palisade mesophyll cells are closely packed columnar cells, and most photosynthesis occurs in the palisade cells. So if you take a look at this picture, what we're looking at here is the dermis. Let's start over here. So on top of it is this cuticle or some waxy stuff generally put in there. Then there's a palisade parenchyma area where these cells do most of the photosynthesis, and then there's a spongy parenchyma area, and then of course there's the lower epidermis, and we're done. In the lower epidermis there's a stomata. Inside, what you're going to see is so the parenchyma is going to make the food has to be transported away. So you need some xylem and you need some phloem to take it away. And you're also going to need a little bit of collenchyma to give the leaf some structure and support. Spongy mesophyll cells are loosely packed with air spaces containing carbon dioxide and oxygen. Vascular bundles of xylem and phloem in the spongy mesophyll appear as veins on the surface of the leaf. So when we look at those bumpy things, long bumpy things on a leaf, those are actually the veins that carry the food and the water back and forth. Venation is the arrangement of leaves of veins on a leaf, and you might not think much of it, but actually it tells you a story. Monocots have parallel venation, whereas dicot leaves form a branched network called net venation. So we'll come to that in a minute, but let's just look at the leaf one more time. This tissue is completely modified for photosynthesis and transporting food. So again, cuticle is the top waxy covering. Then there's the upper epidermis, the palisade layer, and then there's the spongy mesophyll, and the vascular bundle, guard cells. So those are the main features of a leaf. There are no petioles in dicotyledons. There are large petioles in monocotyledons. So this is something that is kind of cool to look at. And also, sometimes the leaf itself is modified into a tendril or a prickle or spine. So these are actually leaves, but they're modified. A stem develops buds and shoots and usually grows above the ground. The stem is divided into nodes and internodes. The nodes hold buds. The internodes distance one node from another. Stems have meristems that generate new tissue. So what is apical dominance? Apical dominance means the main central stem of the plant is dominant over other side stems via the control exerted by the terminal bud over the outgrowth of lateral buds. So if plants have apical dominance, then that allows them to get taller and not so much wider. So here we have a typical stem. Here is a node, here's another node, so this area therefore becomes the inner node. Here's a leaf, here's a bud tucked in, that is the apical meristem where growth occurs. Stems can be modified, so sometimes you have stolons or runners, like here is a strawberry plant, and then this is a modified stem. Instead of going up, it is going to the next plant where it falls into the soil and becomes a new plant. Rhizomes. So rhizomes are modified stems. They have a lot of food in them, and they too can form new plants. So they are not under the ground. They sit on top of the ground, and they are like ginger, iris, and potatoes are also considered modified stems. Angiosperms are about 100 million years old. There are about 250,000 species, but only a few hundred actually are benign. And that is kind of interesting to know because, you know, one would think that with all the plants that are around here, all plants are here for us and they would be very benign, but that's actually not true. It's only a fraction of them that are benign. Most of them are hostile to us. There are two groups of angiosperms called monocotyledons, which I've been talking about all along, and dicotyledons. Dicots are sometimes written in your book as eudicots. So why are we calling them that? Well, monocotyledons have one seed leaf, and those would be the bamboos, the orchids, the palms, lilies, grasses, and grains. So it's a really big group. They just have the one seed leaf when they are seeds. And dicotyledons have two seed leaves, so those have two leaves of food for the baby plant, and those are all the fruits and vegetables that we know of. Let's see how they're actually organized. So if we look at the organization of plants, what we see are monocotyledons are in red, and the rest are all dicotyledons. In the dicotyledons, the examples go from lilies and poppies and roses and sunflowers and tomatoes and magnolia trees and so on. And the monocotyledons are just corn, ginger, lily, orchid, and plantains. So let's look at monocotyledons first. Monocotyledons, we already mentioned they have parallel veins on their leaves. So you don't actually have to wait for the whole plant to make a seed and find out does it have one leaf or what does it have two. You can just look at the leaf. And when you look at the leaf, it'll tell you I'm a monocotyledon or I am not. The stems, if you cut the plant and you cut the stem, you will actually see the vascular bundles are not organized. They're scattered around. And you don't have to cut the stem. You can just look at the petals of the flower. They're always in multiples of 3. So that is kind of cool. The roots of monocotyledons are very shallow. They're fibrous like a mat. And they're more suited for light rain and for sandy soils. So they don't go down deep. Here are examples of monocotyledons: lilies, orchids, ginger, pineapples, bird of paradise, bamboo, bananas. These are all monocotyledon plants. Here are what a cotyledon actually is, or what one seed leaf means. So this, it just has the one cotyledon to feed the growing plant. The veins are all parallel. The flower parts are in multiples of 3, and the vascular bundles are usually scattered around. So here are the leaves, many kinds of leaves, and you can see they're all parallel. Stems have scattered vascular bundles near the outside of the stem, and they're usually surrounded by a large parenchyma in the cortex region, and there are no pith regions in monocotyledons. So here is a corn stem. So what you see is the ground tissue is all around, and the vascular bundles are kind of just, wherever they are. They're just thrown in here and there. Monocotyledon roots have vascular bundles, however, arranged in a ring. So here is the root here coming out. Here are the bundles that are arranged in a ring inside. Here is a more detailed way of looking at it. So again, the outer layer is the epidermis, and what you have is this is all, of course, cortex, filler stuff, and then you have the endodermis with the Casparian strip, and then the pericycle, which will make the new root go this way, and then of course the vascular bundles are inside it. Dicotyledons, they have a multi-branched network of veins on the leaves. So if you look at a leaf and you see nothing parallel but all kind of branched, that's a dicotyledon plant. The vascular bundles are in a ring inside the stem, and the flower petals are always in multiples of 4s or 5s. They have large vertical tap roots, and so they're more suited for deep groundwater. So they tend to be rooted deeper than monocotyledons. Here are some examples of dicotyledons: hibiscus, chrysanthemum, geraniums, carnations, roses, and daisies, and much more. So here are what their main features are. So it has 2 cotyledons. So if you open up a seed, it just has 2 parts to it, with the baby plant in the center. The veins are usually net-like. The flower parts are usually in 4s and 5s, and in the stem, the vascular bundles are neatly arranged in a ring. Dicotyledon stems have their vascular bundles in a ring arrangement surrounding parenchyma cells in a pith region. Between the dicot bundles and the epidermis are smaller parenchyma cells making up the cortex region, as here. So the center is called the pith, and these are the vascular bundles. Again, that would be the epidermis. In the roots of the dicotyledons, they have their xylem in the center of the root and the phloem on the outside. So when I showed you a generic root, I was actually showing you a dicotyledon root. So these are all xylem, and this would be all phloem. All right. And here again, and that's in the dead center of the root. So if we compare the two, monocotyledons, like corn for instance, they have fibrous roots and they're not very deep, whereas a dicotyledon would have a taproot which goes way deep, and it branches. Here is a comparison of the 2 roots in a cross section, and so on the the right hand side, these are monocotyledons, and on the left hand side, these are dicotyledon roots, and what you see is stuff just in this very big center. The rest is not for xylem and phloem. It's just here, and the xylem and phloem form this characteristic X, whereas in monocotyledons you don't see that. You do see a central bundle of vascular tissue, but they're all arranged in a ring, no X. Here is another picture of looking at it, another way of looking at it. Here is a monocot root. So first thing you see is the cortex. The next thing is the endodermis, then it's the pericycle, then it's the phloem, and then it's the xylem, and then there's a pith. In the dicotyledon, there is no pith. There is a xylem in the center, then there's phloem surrounding it, then there's the endodermis cortex, and so that's how they're different. Here's another way of looking at it. So if you look at the vasculature in a monocotyledon stem and a dicotyledon stem, you see they're distinctly different. Just dumped around, and these are organized in rings. The difference between the leaves is very stark. In the monocots, you can see the venation is parallel, whereas the dicot is a network style. Here is a nice summary slide showing the differences between a monocotyledon and a dicotyledon. So monocots have one cotyledon in the seeds. Dicotyledons have two. In the leaf, the differences are stark. The venation is parallel in monocotyledons, but network-like in dicotyledons. The vascular bundles are usually complexly arranged in monocotyledons in the stem, but in a dicotyledon they're arranged in a ring. In monocotyledons, the root system is usually fibrous and shallow, whereas in dicotyledons the taproot is present. It's one long root, and it's pretty deep. The floral parts in a monocotyledon are usually in multiples of 3, but in dicotyledons it's usually 4s or 5s. Another way of looking at the same idea, and I'm bringing it up again and again so that one of these pictures will resonate with you, and you'll take it with you and you'll remember it, because we do need to know all these different parts. So the distinction between the roots and the monocotyledons and the dicotyledons is very stark. Distinction between stems between monocotyledons and dicotyledons, very, very important. The seed, leaf, and flowers are kind of easy to remember, but the root and stem, you need to be perfectly clear on that. And so I brought it again. So here we have a dicotyledon, and here we have a monocotyledon. Because the dicotyledon has floral parts that are in 4s or 5s, if you actually looked at the pollens, the pollen will have 3 pores. But if you looked at monocotyledon pollen, it would just have 1 pore, 1 hole. Another way of looking at the same vascular bundles, very important to remember. And here we see the pollen in monocotyledons. It looks sort of like this. It has 1 sulcus, whereas in dicotyledons the pollen is mostly looks like that, a tricolpate, so it's like a pointy hat sort of thing. About 50% of the dicotyledon species are woody, but monocots tend not to be woody at all. So now we move on to reproduction in angiosperms. The flowers are the only reproductive structures of angiosperms, and they range from being very colorful and conspicuous, such as in a rose or an orchid, to being very simple, reduced, and inconspicuous, such as those of grasses and oaks and elms. The function of a flower is to produce the reproductive cells of the plant. Those would be the eggs and the pollen, and then produce the seeds, which would be the dormant young plant of the next generation. So let's look at the flower parts. So first of all, we'll start at the bottom. Here's the stalk. The stalk is called the pedicel. The pedicel usually has an expanded area here, which is called the receptacle. And that is where the female part sits. But surrounding them are usually leaves called sepals, and then leaves that are brightly colored called petals. On the receptacle will sit the ovary, which has all the eggs, and around the ovary will be long filamentous stalks with the big heads called anthers, which are the male part of the plant. The female part of the plant would be the ovary with a long neck called a style, and a fuzzy top called a stigma. And it's fuzzy because it's going to catch the pollen that is dumped on by air from itself. So the words to remember: stamens. Stamens are the male parts. They have the filament and the anther. The pistil is the female parts of the plant, and that would have the ovary, which is this, the long neck, which is called a style, and then the fuzzy stuff on top, which is called the stigma. The petals of a flower are always called a corolla, and the sepals, or the green part leaves underneath the petals, are always known as the calyx. So here are the male and female parts of the flowers given to you one more time, maybe a little bit better so you can actually see this much nicer. Here is the stalk. Here are the petals, and I'm sorry, the sepals, the petals, and that is the receptacle on which the ovary sits. Around it are the filaments of the male parts, which have the anthers on, and then the female style, which has a sticky top called the stigma. Again, the same idea, shows you the female parts a little bit better, because now here the pollen has been deposited here, and all the pollen is now going to start moving down to the eggs. They're making pollen tubes or eating their way through the style. So parts of the stamen, you should know that the anthers actually have these cells called microsporangium, which form microspores, which are then distributed. The flower parts, so we should know these. The pistil receives the pollen to fertilize the egg located in the ovary, and the stigma receives pollen which grows down the style to the ovary. The ovary contains ovules which contain an egg. A stamen consists of a long stalk called a filament that supports an anther, which is where the pollen is produced. Why do we have petals? To attract pollinators. Why do we have sepals? They cover the immature flower bud and may later help in protecting the seed, or can be colorful and help attract pollinators. And the receptacle is the top of the flower stalk on which the flower sits. Plants can be hermaphroditic, which means they have perfect flowers. Those would be have the ones that have both the male and female plants parts. Those are apples and tulips, for example. They can be called monoecious. Those would have separate male flowers and female flowers on the same plant. So, for instance, corn. They will have male flowers and they will have female flowers, but they'll be on the same plant. Pine trees are like that too. They could be dioecious, and that would be the plants that have male and female flowers on separate plants. And so there's some plants that are completely male and some plants that are completely female. And those would be holly, maple, and saltbrushes. They can be gynoecious. So these would be plants that have only the female flowers. They don't have any other flowers. Those would be, so this is just a division of this. Or andromonoecious, those would be plants that have only male flowers. So I did get you a very nice link, and I would really like you to look at it. Unfortunately, I do not have the software where I can watch it with you. So please accept my apologies for that. Please click on the link and come back to me, and we'll continue on this journey and finish it up. All right, I hope you enjoy it. Okay, so I'm glad you looked at the link. Here we're going to look at how do flowering plants reproduce. So we have a lily over here, and we already looked at the parts of this the flower. What we see is the anthers are going to produce pollen, and this pollen is going to land on the stigma, and the stigma will allow the pollen to move on down. It makes pollen tubes, and the tube will carry the sperm nuclei and it will come all the way down to where the eggs are, and it will find an egg and it will fertilize it. It will dump the DNA component in there, and it will fertilize it, and once it becomes a zygote, then there will be an embryo and a coat will form around it, and that seed will be dumped, and a new seed, a seed will become a new plant, as in here. So here is the actual story behind it. Remember, this was meiosis, and how do we develop the male part? So that's going to be on the left. So the gametes are formed from the anthers inside the microsporangium. Each microsporangia will have the diploid cell, which will then divide by meiosis into 4 haploid microspores. Each of those 4 will develop into a pollen grain, and it becomes mature, and when it becomes mature, it will form 2 sperm, and they will land on a pollen, on a stigma of a carpel, and the pollen tube begins to grow. On this side over here, this is the female side, so that is the eggs. How do they start out? Well, there is a megasporangium area. There's a diploid cell, a megasporocyte, and that would be the mother cell, and that is going to divide by meiosis again, and so it will have 4 haploid cells, but only one is going to survive as the egg, or the megaspore, and then this will be kept. The megaspore is then divided by mitosis, and the egg has a few protective layers of cells around it, and so that is how the egg is kept. This is just showing you the entire life cycle, and remember, anything over here is going to be diploid. Anything under here is going to be haploid because meiosis is going on, and it shows you the same things that we just talked about, but in I think a prettier way. So if you like this picture better, we should just use that one. Vegetables and fruits, so what are they? The seeds of flowering plants are surrounded by a tissue called the fruit, which may be fleshy or dry. The designation of vegetable is actually based on the use of the plant part, eaten as in a meal. So there really isn't a vegetable in botany. We just call it a fruit. But in common terms, for language, we call vegetables the parts that, you know, we're not going to make it into, they're not sweet, they're usually savory, and so we make those distinctions later, but in a plant, there's, generally just speaking, it's just the fruit. Vegetables are actually various plant parts. Some are fruits, like tomatoes and peppers. Some are leaf stalks, like celery. Some are leaf blades, like the spinach. Some are lateral buds, like Brussels sprouts. And some are young shoots, like asparagus. And some are massive flowering structures in the bud stage, like broccoli that we eat. And some are roots, like sweet potatoes. And some are underground storage stems, like white potatoes. So when we call them vegetables, we're actually just eating various parts of the plant. And so they're not necessarily vegetable as such. We rename them as so. What is a fruit? The function of a fruit, of course, is just to protect the seed, because the seed is what we just made. We did meiosis, and we had pollen and eggs, and we made them into a zygote. So that is a new plant, and that has to be dispersed, and it has to be protected. So the function of the fruit is to protect the seeds inside, and to also to aid in seed dispersal. The protection occurs by hardening of the fruit to make accessing the seeds more difficult, so predators don't eat them, or by accumulation of acids or toxins, so they make them taste bad. Fleshy colored fruit attracts birds and animals. And this is done deliberately by the plant. They do try to attract them because they have found out a way to protect the seed so that the seed never gets digested inside these animals. And they use these animals for their locomotion properties and to take the seed somewhere far away. So the seeds will pass through the gut unharmed. Meanwhile, the bird and the animal will get a benefit out of it because they get to eat it. They get to eat the fleshy part, and so everybody benefits. Many fruits promote wind dispersal, so this doesn't take that much energy to do. I mean, no need to make a fruit out of it, spend all that time to do that, but instead just make it really, really small and rely on the wind because there's always wind. Other fruits have hooks, spines, and bristles that readily cling to fur and to clothing. So other plants have other ways of dispersing their seeds. Fruits called pods will dry out as they mature and rip open, and they fling out the seeds. So they burst open, and so that's how seeds can be sent away. If we look at a fruit, what are the structures inside a fruit and how do they actually, what do we call them? Well, there's a pericarp. The pericarp are the layers of fruit derived from the ovary and surrounding the seeds. So sometimes when we eat the fruit, those would be those layers that we're eating. The receptacle is going to be the top of the flower stalk on which the flower resides, remember. The inferior ovary is one that is located within the receptacle. Sometimes it's dipped down. Fruits are structures that are derived from the pericarp, which may form fleshy or hard layers. So when we call it a berry, that is an entirely fleshy fruit containing one or more seeds like grapes and avocados. So that fruit is called a berry. A pod is a dry pericarp that splits into two. So the seeds are usually aligned in a row, like as in a pea plant. Some fruits are called Hesperidiums. This is a berry with a thick, leathery rind and many internal sections that form locules like oranges and grapefruits. Those are really called Hesperidiums, and they are a type of berry. A nut. In a true nut, the entire pericarp forms a single hard dry wall around a single seed. So those would be chestnuts or acorns. We also have fruits called drupes. So again, this is based on the pericarp. So the inner layer of the pericarp, not the seed itself, the inner layer of the pericarp will form a hard layer, and the outer layers may be fleshy or dry, like peaches. So peaches are really drupes. Or an achene. Achene is a small dry pericarp that dries, leaving empty space around a single seed, and those would be buttercup, if you've ever seen a buttercup dry up, or cannabis. Here are examples of, so here's the buttercup. If you've ever seen it, this is what it looks like. That's our drupe. These are true nuts that we see here. And then we have the Hesperidium and so on and so forth. So I just wanted you to have a look. Tissues of some fruit develop from flower structures other than the ovary. So if we have a pome is a fruit where the outer tissue arises from the receptacle, and it's usually discernible from the inner tissue of the pericarp, like in an apple or a pear. Or sometimes we have something called a pepo. The outer tissue is not pericarp, and it forms a tough skin-like outer covering, like the cucumbers and melons. So that is a fruit called a pepo. Or we have aggregate fruits, and they develop from many pistils that are present in one single flower. So if in aggregates you can have a fleshy receptacle, as in a strawberry, the receptacle becomes fleshy and the seeds are in achenes. Or you can have a raspberry where there's a cluster of berries and each ovary forms a fleshy drupe-like fruit. So if you look at the raspberry and the strawberries, they're different. Or you can have an aggregate fruit as a hip, where the seeds and the pericarps form within a fleshy receptacle, as in a rose hip, and the ovary was always inferior here. Here are the pictures that you can see of the same things. So here are the strawberries, look very different from raspberries. If you actually look at them, this is a single drupe. these are the achines, and the flesh has grown up into it. So it's very interesting when you actually look at your fruit in a different eye after having learned all this stuff. How do these fruits germinate? So again, in dicotyledons, we have two parts of this. The food is divided into two parts, two fleshy parts, our leaves, and the baby plant is tucked in between. The baby plant forms a hypocotyl. Once it's in a favorable environment, it will start growing, and it actually forms this arch. The arch will start to move down, and when it moves down, it's called a radicle. But the arch also then gets bigger and starts to move up, and it pulls up the entire food supply out of the ground, and the cotyledons are exposed to the air. And in between the cotyledons, the plant will now form its first two leaves. And the cotyledons have served their purpose. They were there to support the plant as it grows from a tiny hypocotyl with a radicle and baby roots until it has been able to secure itself in the ground and start sucking up food. So the cotyledons then dry off and just shrink, and you start getting foliage leaves. In a monocot, it's about the same idea, but there's only one cotyledon. So over here, when the situation is favorable, the baby plant doesn't quite make a hypocotyl. In fact, it goes straight into making a radicle and something that's going to grow into its shoot. So it goes straight up and it starts making its own foliage leaves like that. So it's a little bit different from a dicotyledon. Alternation of generations one more time. So we looked at this before. Remember, the sporophytes are the generations that we generally see a lot. Those are diploid, and the haploid part gets smaller and smaller in advanced flowers and plants, but is much pronounced in algae. Here is a picture also of the alternation of generations on ferns. We looked at this before too. So please remember the diploid and the haploid parts. And then we go on to nutrition. So how does a plant eat? Well, it must obtain primary macronutrients and secondary macronutrients and also trace minerals for growth. The primary macronutrients, which it requires in bulk, is nitrogen, phosphorus, and potassium. It needs this in bulk. And then there are three secondary macronutrients. It must have calcium and sulfur and magnesium in bulk. Then it needs trace minerals or micronutrients, and those would be boron, chlorine, manganese, iron, zinc, copper, molybdenum, and nickel. If you remember, these guys are all parts of enzymes, and those are just, that would be sodium chloride, so that's part of salt. The macronutrients nutrients are called macro because they're consumed in larger quantities. Micronutrients are present in plant tissue in quantities measured in parts per million. So usually they range from 5 to 200 parts per million. Very, 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 very small quantities. The nutrient uptake from the soil is achieved by cation exchange, where the root hairs pump hydrogen ions into the soil through proton pumps, and these hydrogen ions displace cations attached to negatively charged soil particles, allowing uptake. Soil is always negatively charged, so if you're going to pump in positive charges, you're obviously going to create a current. And the root, especially the root hair, is the most essential organ for the uptake of the nutrients. Nutrient ions are transported to the center of the root, the stele, in order for the nutrients to reach the conducting tissues, which would be the xylem and the phloem. The Casparian strip, which is a cell wall outside of the stele but within the root. It prevents passive flow of water and nutrients and helps regulate the uptake of nutrients and water. The xylem moves the water and inorganics within the plant, and the phloem accounts for the organic molecule transportation. Water potential plays a key role in a plant's nutrient uptake. Water potential is the tendency of water to leave one place in favor of another. Water will always move from an area of high water potential to an area of low water potential, so that's in the plant's favor. If there's a lot of water in the soil, it's just going to naturally move by diffusion into the plant, and the plant actually needs it. So once it's there, then it can lock onto it and start pulling it up. If the water potential is more negative within the plant than the surrounding soils, the nutrients will move from the region of high solute concentration, which is in the soil, to an area to the area of low concentration, which is in the plant. There are three ways plants uptake nutrients through the root. First is simple diffusion. That occurs when a nonpolar molecule follows a concentration gradient, moving passively through the cell lipid bilayer membrane without the use of the transport proteins. Or you could have facilitated diffusion, and here the rapid movement of solutes or ions following a concentration gradient facilitated by transport protein. So this is just hurrying it along a little bit. And then there's active transport, which goes against the concentration gradient. This is the uptake of cells by cells of ions or molecules against a concentration gradient, and this requires an energy source, usually ATP, to power the molecular pumps that move the ions or molecules through the membrane. Many plants engage in symbiosis with microorganisms. Remember, one of the macronutrients that plants need is nitrogen, and plants can't actually take up atmospheric molecular nitrogen, so they have to take it out from the soil, and in the soil usually it's bound in the form of nitrates, which bacteria will produce for them. There are two important types of these relationships. One is with bacteria like rhizobia, and they carry out biological nitrogen fixation in which atmospheric nitrogen, which is molecular, is converted into ammonium. And then they can also have symbiosis with mycorrhizal fungi, which through their association with the plant roots help create a larger effective root surface area. Both of these mutualistic relationships enhance nutrient uptake. And here are pictures of both. So here are rhizobia nodules. So these are actual areas where bacteria are sitting and fixing the nitrogen for the plant. But when you see something like this diffuse stuff, this is fungus, and what this fungus is actually doing, it's not bad, it's good. What it's doing is it's actually making a bigger surface area for the roots, so the roots don't have to build that much of themselves, so they can actually take up more nutrients from the soil. And lastly, there are plant hormones, and we wondered about that. Plants do have hormones. They do need some control and balance, and there are several kinds. So there's auxins. Auxins are for growth and for root branching. So these are good ones. Cytokinin, those are for growth and for making new buds. So auxins are for root branching, whereas cytokinins are for new buds. Gibberellins, they elongate the stem. And then there's something that would obviously have to stop this, and that is called abscisic acid. That inhibits growth. And then there's ethylene, and that is going to stimulate the death of the flower. So at some point when the egg has been fertilized, we don't need the flower anymore to attract any more pollinators, so we need to get rid of that so we can go on to the next stage. And then you need ethylene, which is going to kill the flower off. So here's a chart of the hormones. So gibberellins will increase the speed of seed germination, promote stem elongation, induce early blooming of flowers, and increase the fruit size. And they are usually present in the root and shoot apical meristems and seeds. They're auxins. And they are for stem elongation. They stimulate root branching and they promote fruit development. They're present in the apical meristems and in the immature plant tissues. Then there's ethylene. That is going to increase the speed at which fruit ripens. It stimulates leaf dropping and the death of flowers, and we use that when we take off green bananas from the plants and we want to transport them many days, so we don't want it to die. But by the time it gets to the supermarket, if it's still green, we'll pass ethylene gas over, we'll gas the bananas to make them look green from the outside, because that simulates the death of the flower or any part of the plant. Abscisic acid will inhibit growth and reproduction, and they will stimulate closure of the stomata. So it's everywhere. It's in the leaves, fruit, root tips, and seeds. And then there's cytokinins. They cause rapid cell division. They induce seed germination. They initiate new branches from lateral buds, and they're primarily in roots and fruits. So here is another last table that shows you the plant hormones. These are given right here, and the color shows you where they're used. Gibberellins are used in germination, growth to maturity, flowering, and some part of fruit development, but not for these other parts. And then if you look at auxins, they are not used in germination, but they are used in maturity and growth and flowering, and a little bit of fruit determination and so on. So that is those hormones that we just talked about. Well, let's look at them individually. The auxins are found at the top of the coleoptile, and it is responsible for the plant to be able to react to its environment without having a nervous system. So it's really important because plants don't have a nervous system, but they still need to respond to the environment's stimuli. So the auxins help do that. They elongate the root and stem. They develop leaves and flowers. They respond to sunlight, and they contribute to organ shaping and proper development of the plant and its overall architecture. So these are really good hormones. They help the plant maintain the polarity of its growth. So it knows which way is up and which way is down, and it actually recognizes where it has its branches or any organ connected. So it sort of knows its place in space, in 3D space. So auxins are sort of like the brain of the plant. And the ratio of auxin to cytokinin determines the initiation of root versus shoot buds. So the push and pull between the two will determine how many roots and how many shoots should we have. Gibberellins are involved in the natural process of breaking dormancy and germination. They stimulate cell elongation, breaking and budding, and seedless fruits and seed germination. And if it's too cold, it will stimulate the production of gibberellins. Cytokinins promote cell division or cytokinesis, which is why they're called cytokinins, in plant roots and shoots, and they're found in roots and other actively dividing tissues. They're involved primarily in cell growth and differentiation, but they also affect apical dominance, shoot and root morphogenesis, axillary bud growth, and leaf senescence, which means senescence means dying, so got to get rid of things too. Cytokinins slow aging of plant organs by preventing protein breakdown and activating protein synthesis and assembling nutrients from nearby tissues. The push and pull between cytokinins and auxins. Cytokinins move from roots to the shoots and they signal lateral bud growth. Auxins from apical buds travel down the shoots to inhibit axillary bud growth, but promotes shoot growth and restrict lateral branching. So there's a push and pull, and that's a balance between the two give the plants its architecture that you actually see. Abscisic acid. The abscission means to cut, and it's the abscission of plant leaves. So it's want to get rid of it. So it will slow the plant growth. It will direct leaf primordia to develop scales to protect the dormant buds during the cold season. It will inhibit the division of cells in the vascular cambium. suspending primary and secondary growth. And abscisic acid is also produced in the roots in response to decreased soil water potential. And it translocates to the leaves, where it rapidly alters the osmotic potential of stomata guard cells, causing them to shrink and for stomata to close. So this is a very important hormone, and it regulates how much loss the plant can endure during the day from its stomata. Ethylene is also a hormone. It's used for ripening fruit. It acts at trace levels by stimulating or regulating the ripening of fruit, the opening of flowers, and the abscission or shedding of leaves. It's been known forever. It's been used by ancient Egyptians and Chinese for millennia. And then we have tropisms. Tropisms, what does this mean? These are also the reactions of plants to the environment. Turning in response to a stimulus from the environment is called a tropism. Tropisms are positive. When they're positive, they're towards the stimulus, or they're negative if they're away from the stimulus. They can be chemotropism, which means movement or growth in response to chemicals, or gravitropism, which means movement or growth in response to gravity, or heliotropism, which is movement or growth in response to sunlight. Sometimes you put the plant in sunlight in a window and it starts to grow towards the sunlight. That's heliotropism, or sonotropism, which is movement or growth in response to sound. And yes, you can do that too. Hydrotropism is movement or growth in response to water, and we see this a lot. Phototropism is movement or growth in response to lights or colors of light. Thermotropism is movement or growth in response to temperature. And electrotropism is movement or growth in response to an electric field. Thigmotropism is movement or growth in response to touch or contact. Many plants, if you touch them, they will close up their leaves, and that is called thigmotropism. A phytochrome is a photoreceptor. It's a pigment that's used by plants to detect light, and these are sensitive to light in the red and the far red region of the visible spectrum, and plants will use them, flowering plants will use them to regulate the time of flowering, like how many days should we be flowering based on the length of the day and the night. So they calculate the photoperiod and to set circadian rhythms. So they do have circadian rhythms just like you and I, and that is caused by a photoreceptor or a thinking sort of cell, which is called a phytochrome. The photoperiod is the length of the night, which is the controlling factor in sensing seasonal changes, and that is used as a signal for the flower. Photoperiodic flowering plants are classified as long day plants or short day plants, and each plant has a different length, critical photoperiod or a critical night length period, so it knows how long to stick around and become a flower. And with that, we come to the end of the course. Congratulations, students. We finished biology together. I've enjoyed having you, and I hope this was helpful.